We are going to get into cladistics after our discussion on taxonomy and systematics and naming of things. It makes sense to take a look at how these things are related through homology and analogy. The first thing we need to talk about is homologous versus analogous characteristics or characters. Homologous characteristics or homologous characters are those that are resulting from common ancestry. This means that they have similar structure and embryonic formation. Thus, they're often used for classification because they're a good, reliable source of information regarding the relatedness between two things. The flip side to that coin are analogous characteristics or analogous characters. These analogous characters have similar function, but they are not similar structures. They are usually not used to classify organisms because they don't represent something shared due to common ancestry. We also call analogous characteristics homoplasy, and we'll talk about specific examples of homoplasy in the coming slides. The best example that we have for homologous characteristics are wings in birds and in bats. And this is an example of homology. The bird wing and the bat wing are homologous structures, meaning that they share a common evolutionary origin between the two. And it just so happens that they are used in the same way. Something that is an analogous character is the set of wings that insects use. So the insect wing is analogous to the bird and bat wing because even though it's a wing and it's used for flying, it is not derived from common ancestry to those organisms. An example of something that is homologous that you may not really think about or appreciate are the bones in our inner ears. So here we're talking about homologous bones. They share common ancestry, but they're used for different things. So here we're talking about the different evolutionary trajectories of the middle ear bones in mammals. We have in our heads the quadrate bone, the articular bone, and the stapes bone. These tiny bones in our ears and mammal ears, they help us hear. But what's so fascinating is that these same bones are used by reptiles for chewing. So we can take a look at the same homologous bones that we have in our middle ear, here. So the articular, the quadrate, and the stapes, and for example, the stapes here is colored in green. These are used in our reptile friends for chewing. So the reptilian quadrate and articular bones are homologous to the bones that we have in our inner ears called the incus and the malleus bones. This is an example of divergent evolution where the same homologous bones are actually used for different functions. In this case, for hearing for mammals, and then chewing in the reptiles. There's more to learn about homologous and analogous structures in the TED Ed video that I've set up for you, and specifically a lot to do with the bones that are in our forelimbs, and then forelimbs of other organisms. So let's talk about, instead of divergent evolution, convergent evolution. Con means coming together, and you can think of convergent evolution as a reoccurrence of form. Convergent evolution is a process whereby organisms that are not closely related, in other words, they're not monophyletic with each other, they independently evolve similar traits as a result of having to adapt to similar environmental conditions or ecological niches. An example of this is the torpedo-shaped body of a fish compared to a swimming mammal like a dolphin or a whale. The first mammals were actually terrestrial and they walked on 4 limbs. And what's interesting is that the ancestors of the cetaceans, which are dolphins, they adapted to swimming and their limbs were reduced and their body shape converged on the typical fish shape. So because these two organisms are living in the same type of aquatic environment, it makes sense that they would want to have similar body plans like this torpedo shape that allows them to zoom and zip through the water. It does not mean that the character of that torpedo shape is derived from a common ancestor. We can talk about homoplasy and analogous features, and this is when morphology does not equal homology. As in the previous slide, that is an example where morphology does not equal homology, and we call this state of being homoplasy. So that torpedo-shaped body in the previous slide, that is an example of homoplasy. And homoplasy is a character shared by a set of species, but not present in their common ancestor. So what we just took a look at is an example of homoplasy. We can also take a look at another example that has evolved many, many, many times, and that is the convergent evolution of the eye. The evolution of the eye actually occurred many times independently in many different species across the vast tree of life. So we can think of things like octopi or ray fin fishes, things so divergent like a crocodile and a hippopotamus. They all have eyes, but it's not necessarily derived from a common ancestor. Instead, this is a relationship because of convergence or convergent evolution, where it just made sense in that environment to evolve the ability to see. And that's why we see this character trait of having an eyeball arise so many times across the evolutionary tree of life. Another example, getting back to body shape, is the example that we have between the Tasmanian wolf and the coyote. Now, you may not have heard about the Tasmanian wolf before because it's actually an extinct carnivorous marsupial. And we haven't had these around for, I guess, close to 100 years now. But just taking a look at their body plans, overall, they look very, very similar to each other, but they are completely separated on the evolutionary tree. We can just break down in a table what characterizes a homologous structure and an analogous structure. So our homologous structures, they arise through shared ancestry. They have similar internal structures, but they may look different overall. Our analogous structures, they arise through no shared ancestry, but they serve a similar function, perhaps for swimming very quickly in water or flying through the sky. And therefore, they may look similar externally. We have different types of characteristics or characters that we use to build our phylogenetic trees or our cladograms. These different characteristics or characters can be broken down in this way. We have some characters that evolve pretty slowly and others evolving rapidly. This is what we call mosaic evolution, where you have some genes that are more prone to selection and adaptation and others that are pretty set in stone evolutionarily. Then we have ancestral characters. These are older forms of traits. These are the types of characters that would be found in the last common ancestor between two groups. And then finally, we have derived characters, things that have arisen more recently in geological time. This is newer forms of traits relative to other organisms, and these serve as really good markers for discerning evolutionary lineages. We can compare traditional versus cladistic systems. So I've used this term cladogram before, and we're going to get into on the next slide what that actually means. But here we have traditional evolutionary systematics using phenotypic or the physical appearance of something to determine similarities and differences. This is called a phylogenetic tree, and it's based on distinct body plans and an entire way of life. It may not include all ancestors. By contrast, We have cladistics, which uses only evolutionary relationships. This groups all species that share a derived character, a newer character. So for example, a cladogram is going to express the presence or absence of different traits within a group. For example, we would be able to separate the mammals from other groups because we know that mammals possess hair, they possess mammary glands, and they also have a reduction in the bones in the lower jaw. So those are all derived characteristics of mammals that separate them from other organisms. By contrast, we can talk about ancestral characters like having 4 legs or having a spinal column. These are often not very informative characters because they are ancestral, meaning that most things on the tree of life, if we're talking about animals, possess these characters. So they're often ignored when we're trying to build cladograms. And of course, when we put all this together, it's called a cladogram. So what does this look like? Cladograms give a hypothetical picture of the actual evolutionary history of the organisms. And that's here on the right. We have an image of a cladogram taking a look at the different relationships between our amphibians, our mammals, turtles, lizards and snakes, crocodilians, and then finally our birds. Notice these spokes are pretty uniform. They follow a scaling law along this long branch here. We can't really discern too much information based upon the length of the branch. And we'll talk about the different ways to discern relatedness in cladograms in the coming slides. By contrast, phylogenetic trees give an actual representation of the evolutionary history of the organism. So here we have a phylogenetic tree with these same groups of animals represented. We have here at A, the common ancestor of reptiles and birds, and we can follow that through to see when we had the evolution of birds, who the birds are most closely related to, what different branching off points we have where organisms are now extinct and therefore not represented on Earth today, but we do have fossil evidence of them. We could take a quick look at a cute example and try to understand the relationship of parrots to our pterosaurs and our dinosaurs. This slide groups our parrots and pterosaurs together because they have wings. And you know, if you're going off one morphological character alone, that might make sense, but it's actually incorrect because the T-Rex and birds share a common ancestor in reality. So the relationship between the bird wing and the pterosaur wing is an example of homoplasy. What this tree should look like is this. This is the accurate or correct evolutionary interpretation of the relationship between parrots, dinosaurs, and then pterosaurs. To make a tree, first we need to look at all of the patterns of relatedness that are suggested by morphology. Once we have our matrix of the different characters and traits across all of our organisms, then we build a tree. We build multiple trees representing different hypothetical outcomes of relatedness. And we select the tree that suggests the smallest number of evolutionary steps. This is most likely the correct one because of something called parsimony, or the simplest answer is usually the correct one. This is also known as Occam's razor. Here we have a lovely compilation of images of different types of organisms, very different from each other. We have everything from bearded dragons to a Labrador to a turkey. So the question is, how do we make a family tree of all of this? What we want to do is identify clades or groups of organisms that are closely related. we can build these clades by thinking like a tree. And this is, if you're not familiar with it, the very famous sketch that Charles Darwin made in his notebook right before he published On the Origin of Species in 1859, where he traced out the hypothetical relatedness of all organisms on Earth. And he wrote here, I think. So this was just his hypothesis. He had no knowledge of molecular genetics. We didn't even have a field of genetics at this time that he was postulating this idea of a tree of life. I'm in love with this image so much, I got it tattooed on my back. But here, we need to think like a tree. And so we can represent this in different ways. Something that looks more like a cladogram here or a phylogenetic tree here. And let's just walk through an example of what this looks like. Okay, so here we have a whole bunch of beetles with time represented along this y-axis here, going up and down. So the oldest beetles are at the bottom. We see that we did have some mating events, which produced progeny that went off to different locations. And here, at some point, there was some sort of physical barrier, okay? Perhaps a geographical barrier. Maybe there was a river or a stream that opened up and physically separated these two populations of beetles from one another. We'll get more into the mechanisms of speciation in future lectures, but just as a preview, this is what we call allopatric speciation. This is most often what we see in the geological record. All right, so from our original population of beetles here, all identical, nearly identical, we see that they're all green. They produced their progeny, which then mated with each other. There was no cross-mating across this geographic barrier, and eventually these organisms got isolated from each other, so much so that we have in our most recent population different species of beetle. Some look similar to the original batch here, but we do have distinct populations of those that have different physical traits, like blueness or black dots, for example. So if we were to make this into an evolutionary tree, it would look something like this. So we have the common ancestor to our two groups with a splitting off or a branching off because of this geographic barrier in the middle, yielding our white branch here on the right and our green branch here on the left. Let's break this down into some components. We have the green branch and the white branch. Each of these we can call sister taxa. So taxon A and taxon X are sister taxa to each other. If we were to track this population on the right a little bit farther in time, we'll see that there's another speciation event caused by another geographic barrier being introduced and physically separating these two populations. So again, what would this look like on our tree? It would look like this. So now our branch on the right has extended a little bit in time. And because of that geographic barrier, we now have a fork in the road. Taking this back to our example before, we have taxon A, taxon X, and then new taxa B and C, with taxon X representing the common ancestor between sister taxa C and B. We can call this spot on the tree where two branches converge a node. So anywhere there is a V or an L shape on a tree, that is called a node. We refer to these sticks as branches of our evolutionary tree. And then finally, each of these letters is representing either a species or a taxon. We could be talking about things at the genus level instead of the species level, or even at the order or family level. You will remember monophyletic groupings include the ancestor and all of its descendants. So here circled in blue, we have X, C, and B. So this is the common ancestor to B and C, meaning it is the ancestor and all of its descendants. Therefore, we have a monophyletic group. And this, put back on our beetle evolutionary history, we would call monophyletic group X. So B and C are sister taxa to each other. But what about A? A is sister taxa to both B and C equally. Okay, so remember that. They are separated only by one V here. So A is equally as related to B as it is C. And phylogenetic trees show the relationships between different species or taxonomic groups, as we just saw. So it can be depicted as in the previous slide, where it looks more like a ladder model, or it can look more like a tree model, as we see here. We have tips representing taxa that exist today. We also have these dots representing the nodes. You can recognize nodes because they fall in between where you have the splitting off of branches, which are represented here and here. And then finally, you have the root or the stem of your tree, which is usually where you have the last common ancestor. And it's also good to root your tree with an outgroup, and we'll talk about that in the lab lecture. So nodes represent the last common ancestor between two species. And two species or groups that share a node are called sister taxa. That is how we identify sister taxa. So here again, in the black dots, those are our last common ancestors for the groups that are springing forth from them. So let's do a little exercise. Okay, so A and B are sister taxa. Great. D and E are also clearly sister taxa. So C's sister here is both A and B together because they share a common ancestor down here at this node. So the question, D and E's sister is what? The answer is A and B and C. And you'll notice that A and B are inside parentheses inside my answer here. And that's because A and B are as equally evolutionary related to D and E between them. So A is not any more closely related to D and E than B, and B is not any more closely related to D and E than A. They are equally as related to D and E between each other. By contrast, C is a sister taxa, but it does not share the same evolutionary distance that B and A do to D and E. So what is F's sister taxa? F is way over here. The sister taxa at this level would be A and B being equally related, then C, then D and E being equally related. So all of this on this tree is the sister taxa to F. But not all of these taxa have the same evolutionary distance to F between them. Now, Who is most closely related to D? That should be pretty obvious. The answer is E over here. Next, ask yourself, who is most closely related to D and E equally? The answer is that A, B, and C are equally closely related to D and E. And that's because they make up this entire branch over here. So the trick is finding the common ancestor, which is represented at this node, and then everything that springs forth from that split is equally closely related and therefore representing the sister taxa. Next, who is most closely related to F? That's all of these. We have B and A equally closely related, then C, then D, and E. The thing to worry about, though, is that you can switch arbitrarily the positions and orientations of the branches. As long as that node doesn't move, you are free to switch this and rotate it as much as you want. It still represents the same evolutionary distance because what matters is where the last common ancestor is located. So the question is this image the same as the tree that we were looking at before, where we had A and B equally closely related to each other, then C, then D and E equally closely related to each other, and F. So just take a look if this is at all representing a different evolutionary tree than the previous slides. The answer is that this is the same image. It's representing the same evolutionary distance between the taxa. Even though we rotated the branches, it is still representing the same information. What about this one? The answer to this is that it is not representing the same thing. We have changed positions of D and C, which alters the interpretation of relatedness. So now that we have gone through how to read these trees with letters as our taxa, let's take a look at how this would play out with actual animals. Here we have a tree that represents all of our tetrapods, so things that have 4 limbs, including our frogs, our kangaroos, our donkeys, our geckos, our crocodiles, and our aves. The kangaroo, is it more closely related to the donkey or to the frog? So here is our kangaroo, here is our donkey, and here is our frog. In order to go from the kangaroo to the donkey, we just have to make 2 steps, okay? So moving from kangaroo to common ancestor to donkey, 2 steps. Now, how do we go back to the frog? That's more steps. So kangaroo, down here to the amniotes, to the tetrapods, finally to the frog. So that's 1, 2, 3. That's 3 steps. And before, to go to the donkey was just 2. Therefore, the kangaroo is most closely related to the donkey. Next, regarding the crocodile, is it more closely related to the bird over here or to the gecko? We can do the same tactic and just count our steps. To go from the crocodile to the bird, we go 1, 2, so 2 steps. Now let's move to the gecko. We do 1, 2, 3 steps. So again, crocodile is more closely related to the bird than it is to the gecko. What about the gecko? Is it more closely related to the bird or to the donkey? What do you think? Let's go from the gecko to the bird. 1, 2 steps. We don't have to stop here because we're not moving up this branch. Okay, so 1, 2 steps to go from gecko to bird. And then finally, to go to the donkey is 1, 2, 3, 4. It is 4 steps. So our gecko is more closely related to the bird than it is the donkey. We can call the crocodile and the aves, or our birds, sister taxa because they share a common ancestor just here at this node, which is called the Archosaura. Likewise, we can group the crocodilia and the aves together and say that the crocodilia and aves are sister taxa to the geckos, the Lepidosaura. And then finally, if we just wanted to ask what is the sister taxa to the frog that we have, the Lissamphibia, we would have to group all of these organisms, everything from the marsupial to our bird, in order to get the sister taxa for our frog. We can also do other groupings and determine that our donkey and our kangaroo are sister taxa to the gecko, crocodile, and the bird. That was a lot of information. There's supplemental videos and readings for you to do on the Blackboard to really develop these ideas and practice them on your own. We have a pretty intensive lab activity that is all online. You have 3 assignments to do within this module called What Did T-Rex Taste Like? The first couple of assignments just introduce you to the idea of cladograms and interpreting them. And then the special assignment at the end is going to test that you know how to read a cladogram and determine evolutionary relationships between them. So please go to the lab Blackboard before lab and complete folders 1 through 3 from the module and complete the pre-lab quiz that's going to test that you know what you're doing. It should take you about 45 minutes to an hour to complete folders 1 through 3 on this module. And in class together, in lab, we're going to do folders 4 and 5 together. And at the end of the lab, you'll turn in the special assignment either as a photo that you draw or as a PowerPoint presentation that you put together, because sometimes it's just easier to draw sticks and put text boxes in PowerPoint than it is to draw it. I totally understand that. And it is not going to be due until the end of the week at 5 p.m. So I will give you a little bit of grace in finishing that assignment. There is also a lab lecture that I'm going to walk through and post on the lab Blackboard, and it'll again tell you how to read cladograms with a silly example.